Alrighty, good morning everybody. We're going to continue on with our 10:30 session. So each of these speakers does have 10 minutes and then 5 minutes for questions. And our first speaker is Kit Lewers, who's going to be talking to us about exploring harmful algal blooms in the Caloosahatchee. Okay, all yours.
Also, I don't know what happened. That's supposed to say river, but it says Rhode Island. So there's that. Also, I just ran and I'm not an in shape person, so give me 1 second. Sorry. Okay, hi, my name is Kit. This is actually probably going to be a pretty short presentation. This is a class project that I did, and it was originally supposed to be a poster, but I'm just going to talk about it, and there's a lot of failure in it, but that's okay. It's okay to talk about failure. So what is a harmful algal bloom? So harmful algal blooms are typically when algae just overgrows in I'm really out of shape. When algae typically overgrows in rivers, lakes, they happen in Colorado, they happen in the ocean, they happen in freshwater. But typically what happens is it can cause a lot of issues because these algae can release toxins that can impact both people, animals, the river. And where I'm looking at is in Fort Myers, Cape Coral area, Florida. And that's special to me because I grew up in Florida and I spent a lot of my time in Cape Coral. I'm not from Cape Coral, but my mother is from there and my grandparents still live there. And what's super interesting about it is it has what I like to call one of the most useless canal cities in it. So it is really cool, but a lot of these canals do not connect. They just you can go in certain neighborhoods, but there's a lot of blank areas, as you can see, where there's just rivers and they just didn't have them connect, which I think is interesting because when I was a little kid, I was like, it's a canal city like Venice. And then I grew up and I was like, you can't use it like Venice, but it's still pretty cool. So what happens here is in these really small areas this is a picture of a canal that's not too far from my grandmother's house. They get really saturated with this harmful algal bloom because they're only like 5 to 10 feet across, depending on where the canal is. They're super small, and people live here, and their dogs jump into rivers, and people go fishing, and it causes a lot of issues when there are these large algal blooms happening. So what I did is I was like, okay, well, I know that these are big, and not a lot of other people know that they're big. So I'm going to see what we can see using NDVI and satellite imagery. So I used Landsat 7 and 8, and then I used Sentinel data to look for harmful algal blooms from 1999 to 2021. I'm not nervous, I'm just really out of shape. I'm so sorry. Okay. So the first ones that I looked at were Landsat 7 and 8. This was built by NASA, but is now stewarded by USGS. In Landsat 7, there are 8 bands. In Landsat 8, there are 11 bands. And really the only difference is there are 2 thermal bands in Landsat 8. And there's also a coastal band and a cirrus band that is not in Landsat 7. So the resolution is, if you look in the panchromatic bands, so black and white, you can get down to 15 meters. All the other bands except for thermals are a 30 by 30 meter pixel resolution. And it has a temporal resolution of 16 days, which means that it goes around the Earth every 16 days and takes pictures. And then for the later ones, which I was really excited about, was Sentinel-2, and that's operated by the European Space Agency, and it has 12 bands. Also, if anybody does use remote sensing, I created a software package where if you just put in either the band name or what micron or nanometer measurement you're using, it'll tell you where that falls in each band in Sentinel, VIIRS, MODIS, and Landsat. So if you're interested in that, feel free to ask me about that after the presentation. The resolution is if you're looking in red, green, blue, and NIR or near infrared, you can get down to 10 meters. And in the red edge, which is really good for looking at vegetation health, you can get down to 20 meters. And you can look at coastal water vapor and SWIR in 60 meters. SWIR, cirrus only, so there's 3 SWIR bands in all. And each satellite, so there's Sentinel-2A and 2B, goes around the Earth 10 days, but they are placed parallel to each other, so you actually get a 5-day temporal resolution, so every 5 days you can see what's going on. Okay, and then another note, so Florida is very rainy and there's a lot of storms, so I just put in a cloud masking algorithm so I wasn't misclassifying anything that should have been vegetation in the water as just like pure water, because it does kind of like mess with like near infrared and sphere bands when you're like working with clouds. So that is just a correction that I felt was really important to include. So then I ran into my insurmountable challenge. So I was like, okay, I want to use the highest resolution one, and I know for a fact, because I was there, that there was a harmful algal bloom in 2018 and 2016. And there was only one picture from both of those years that I could find something, so I don't know if you can see my mouse. This is an island over here. This is part of a harmful algal bloom that I was able to pick up, and I was like, okay, great, I can see it in the river. Now let's go look at the canals. This is so exciting. I didn't really take into account what the pixels would look like when you're doing 10 by 10 meters on like 5 to 10 foot wide canals. So I wasn't able to use the data, which was really sad. I like looked at this area because I was able to find a picture. This is a marina that's like kind of close to where my grandparents live. And I knew that they had a bunch of algae on these dates. And I was able to see like a little bit of red here, but with the median pixel count, I just couldn't get really any clear information if I could find harmful algal blooms. So for the class project, I'm not going to talk about this here, but I ended up looking at thermal imagery. I just thought this was a really pretty picture. This is a stretched thermal image from Landsat 7 in 2012 of the Caloosahatchee River in Cape Coral. And it's interesting, you can like see all of the little like canals here, like the cooler lines, and then the Caloosahatchee River is nice and bright and going out into the ocean over here. Excuse me. And then I did an urban pixel classification from 2000 to 2023, and we saw a lot more urban pixels pop up from there, but neither here nor there, since this is a 10-minute talk. But if you're interested in this, please feel free to ask me. It was a lot of fun. So that was where I was kind of like, okay, so these harmful algal blooms are happening in like kind of these really small areas. And there are satellite products that you can use to get to a much finer resolution, but I specifically wanted to find vegetation. So Sentinel-2 was really my best bet for that. And they are coming out with more products that are coming to a smaller resolution, but I was like, what are some things that we could do like in the intermediary while things are being built, so we can still kind of keep an eye on what's happening in these river systems and these lakes where harmful algal blooms are becoming more prevalent. So I was like, small scale does not equal small problem. Like probably a lot of you have not heard of the Caloosahatchee River, but there are over 1 million people who live around it. It is really important to Florida tourism. It's really important to Florida health for people who are living in that area. And it is connected to Lake Okeechobee, which is in Central Florida, and a lot of runoff happens there. So there's like, and what's super cool, in my opinion, is the Caloosahatchee River also runs down to the Everglades. So it's very intertwined in this estuary system. And I looked on Florida Fish and Wildlife. I looked on Neon Biorepository. I looked on Neon, even though I knew that they were pretty far from the like Disney Neon Avers pixels. I like couldn't find any data easily. And that kind of concerned me because I was like, I know that this is happening, but other people don't know that this is happening, and I only know that it's happening because I'm there. And like, it makes the news and everything, but there's nothing like quantifiable that I can find on this. So what are some things that we can do in the meantime? I know that Florida Fish and Wildlife takes samples. I've talked to people. I just could not find any information. So one thing that would be really important is making these small scale data more accessible. So more fair, findable, accessible, interoperable, and reproducible. In situ sensors would be really nice also. We have a lot of those in lakes in Colorado that are super helpful for monitoring some algal blooms that seem to be happening with the hotter summers. And then my favorite part is drone photogrammetry. This is a mission that I flew in Roosevelt National Forest a couple of months ago, and this is a digital elevation model, and I was just really blown away by how easy it was to process this data. And I can see like stones in the stream. I can see I can see old buildings that are dilapidated in this digital elevation model. And then when I actually get to the pictures for the RGB, like I was just really blown away by how easy it was to see things. And food for thought, So if any of you know what AVERS imagery is, it is airborne imagery from planes that is taken by both NASA and NEON and a couple of other scientific organizations. Those pixels are typically 5 by 5 meters, and this is what a 5 by 5 meter drone image looks like from the data that I had. So you can still see like there's trees, there's grass, and then just to put it in context of what I was looking with at Landsat, that is a scaled picture of what my drone imagery looked like, and I can clearly see that river, sand, trees, and snow. And if that was on Landsat, that would just be like one colored square for the median colors of what it is. And there's just a lot of detail that's lost. And this is a very cheap and fast way, honestly, to collect data for small areas. And I didn't put this in there, but also just like for reference, the drone imagery that I showed, it was 1.62 centimeters per pixel versus 30 meters per pixel. So yeah, if you have any questions, please feel free to ask me, and thank you so much for your time. And I'm sorry that I was out of breath at the beginning.
Thanks, Kit. Okay, any questions for Kit?
Hey, Kit, that was an interesting talk.
Thank you.
One quick question, well, two quick questions. One is, what's the name of your software package?
Okay.
The other one is, did you consider doing any unmixing of those pixels that didn't have the good enough resolution?
I thought about it, like full disclosure, this was a class project, and I was like, I don't know what I'm doing. So after the fact, I did think about it, and I am still thinking about doing that over the summer, just to make sure that I'm keeping up with my remote sensing skills. And the name of the package is Hyper, so like hyperspectral, because I'm also bringing in all the neon bands to like convert over to satellite imagery. It's called Hyper #2 Leo, and it's on GitHub. I'll share it in an email, or yeah, I'll email you about it. Yeah, I'm working with Bridget on it actually. She's been helping me a lot with it. And John too.
Hi there. Thank you. This is really wonderful and interesting.
Thank you.
I also do, I think, a little bit of similar work, and one of the things that I'm most frustrated with often is missingness in data. I think we can all kind of relate to that. How significant was missingness in the various products that you used, and how did you overcome that for like a time series analysis?
So I couldn't do anything with NDVI. So what I had to do is I tried to see what river temperature looked like, and then what surrounding urban temperature looked like, and see if there were any correlations. And I couldn't find anything. So I really didn't get to make any conclusions about the harmful algal blooms, which kind of stunk. So.
Hi, thanks for your talk. I was just wondering if you know in that area what kind of the average size of an algal bloom is compared to the grain of the imagery you're getting?
So it depends on how big the body of water is. So like I said, the canals are typically only like 5 to 10 feet wide. So they do saturate the entire like canal system, which is why I was also surprised that I couldn't really see it. But like the peak algal blooms did not happen when either Landsat or Sentinel went over that area, was the other thing. So there's a big missing chunks of time. And there were a couple that I even tried to look in tier 2, and the imagery just wasn't good enough. So yeah, there's just like, I knew there was data there, but not that I could make any sense of.
We're going to have to move on.
Okay.
Randy,
you can ask me later.
Ask her afterwards. All righty. Moving on to our next presenter. Caitlin Bloomer is going to talk to us about combining museum collections and predictive modeling to survey a cryptic taxon.
Thank you all for being here today. I think my talk is long, so I'm going to start immediately. So I'm currently a postdoc with the Illinois Natural History Survey. They are part of the University of Illinois in Champaign, Illinois, and I specifically work in the crustacean collection. So we've got over 135,000 lots, over 400 species. It's a really cool collection. Specifically, we have a lot of freshwater crayfish. The last 2 curators over the previous 50-ish years have really specialized into crayfish and shrimp. So they're really well represented, and we do provide all of our specimen data publicly available. Most of our collection, about 50%, is from Illinois because of where we're based, but we do have specimens from across North America. I work on a specific group known as burrowing crayfish. They are such cool animals, and they also suck to sample. Basically, they have a very limited sampling time frame. They're only really active from about March to May every year. That's when you have to go out to get your hands on them. If you want to get your hands on these critters, we have checked a lot of other ways. Repeatedly, it has shown that the most effective sampling method is to get on your hands and knees, stick your bare arm in the ground, and try and grab them. That, combined with the fact that they have got really cryptic burrows, that's kind of the only indication that you've got burrowing crayfish in the area, is like this photo in the middle. They kind of push up dirt from the ground when they're excavating their burrows. And you'll see the photo on the right. I've got an arrow pointing to what a burrow can look like in a prairie. Really difficult to sample, really difficult to spot the burrows, and overall this has kind of culminated in a real lack of distributional data. So whenever we are trying to understand the distributions and ranges of these species, we have to turn to some different methods. One of those that several of you are probably familiar with is ecological niche modeling, also known as species distribution models, habitat suitability models. These are just predictive models that are able to correlate the presence data, like that we have in our museum collections, with environmental data, and they can suggest suitable sampling sites or project to new areas that have unknown distributions. The niche models that I use are through a GUI called Maxent, which is really user-friendly and pretty popular for these kind of models. Now, the big issue that I just mentioned is that paucity of distributional data. I was working on 3 species. From left to right here, we've got prairie crayfish, digger crayfish, and painted devil crayfish. These are 3 of the most widespread and common crayfish species in the United States, and yet whenever I collated all the distributional data I could, not just from our museum, but from museums across the country, state agencies, even the literature, I was still ending up with only between 2 and 6 records for every 100,000 square kilometers of these species range. Really sparse data that we're working with when we're trying to model. So in order to understand their distributions a bit better, to try and solve this problem of where do we survey for these animals, I decided to combine all the digital records for all 3 of these species together from over 50 museums, agencies, and the literature into one multi-species model, and use that to try and find these burrowing crayfish. I'm not going to get into like the nitty-gritty of model evaluation and selection. You're free to ask me about that at any time, but I'm more interested in presenting results and why you should all care about this. So I'm just going to jump straight to my model output. If you're not familiar with what these model outputs can look like, black dots represent all the historic distributional data that we had for all 3 of these species, and the background is habitat suitability. So this kind of blue, the cooler colors indicate low habitat suitability, moving up through those warmer colors into higher relative habitat suitability. And it kind of makes sense when you look at this map. You'll see that the warmer colors do tend to accumulate around areas where you've got more distributional data from historic records. But there are some areas like up around the upper edge of the Michigan Mitten or suitable habitat over in South Carolina that's predicted despite the fact we didn't have distributional records. So an interesting output, and we have to make sure that we go look at our model output to make sure that it is correctly predicting suitable habitat. We can't just take it for granted that it's doing that. So I went ahead and field validated this model in Missouri. This is the area where these 3 species have the most overlap in their range. I selected 98 sites across Missouri, ranging from what was predicted to be really good habitat all the way down to what was predicted to be completely unsuitable. And we did have good success in finding all 3 of our species and getting really, really muddy. And this is a map, that same map just zoomed into Missouri of where I sampled. The white dots all indicate areas that I had detections. The black dots are areas that I did not have detections. And I found that this multi-species model was a significant predictor of burrowing crayfish presence, not only for our 3 species, but I also found non-target species. And what I mean by that is species that are burrowing crayfish, but that I used no historic records to train this model. We weren't expecting or trying to find these species, and we still found them with this model. It seemed to be able to identify kind of broadly suitable habitat for all of these species together. So while our burrowing crayfish do still have kind of individual niches and individual habitat preferences, there is some sort of Venn diagram overlap that this model was able to identify and lead us to find many different species. Now, why this is really interesting is that multi-species models in the past have mostly been used for sister taxa, or very closely related taxa. The 3 crayfish that I was using for this modeling project are all from 3 different genera, and burrowing behavior has evolved multiple times independently across the crayfish phylogeny. So it's very exciting that we might be able to use these multi-species models not just for sister taxa, but for other species that are maybe not as closely related. There's a couple reasons this could be really useful for conservation. First is where we're directing surveys in areas that have few historic records. What I've got up here is a map from our website, the American Crayfish Atlas, and you're looking at all those different colored dots represent different species of burrowing crayfish and the known distributions we have for them across the United States. I've got big black circles around areas where there are historic records all around the periphery, and yet basically nothing within those black circles. It feels really unlikely that all burrowing crayfish looked to Iowa and went, we're good, no thank you. And way more likely that just nobody's really surveyed Iowa for burrowing crayfish. So whenever you're looking at that, like a whole state that we don't have distributional historic records from, you've no idea where to even start looking for these species. And being able to combine multiple species from the periphery of that area to start directing your surveys is a really great way to help your field biologists know where to go and save resources. Another example here is where there are species that are lacking historic records. Like probably a lot of groups that you're working with, genomics is getting better and we're undergoing a lot of taxonomic revisions. The example I've got up here is Leuconicambarus diogenes, or the devil crayfish. This is a species that previously we thought this entire purple range was one species, and it is now 11 or 12, maybe more, we'll find out. Just from Illinois, where I'm based, we have 4 or 5 species from this species complex. So again, we've got all these historic records, and we now have no idea what they represent. So whenever we're using the historic records to build models, being able to just combine multiple species into one and continue directing our surveys within the state is a really useful way for saving resources. Now, kind of coming full circle back to the crustacean collection, all of those historic records, we have 534 lots of this devil crayfish that can now be re-identified. Rather than having to go back to all of those historic records and find out what now lives there, we're able to rely on the efforts of past scientists who have already surveyed the area. This reduces the time, the resources, and the funding that is required to document these distributions. And it allows us to have a much better idea of what the current distributions are of these newly described species while continuing to direct our field surveys to new areas. So ultimately, we're at natural history collections supporting ecological research, not just with their digital data, but also giving a strong shout out for the specimens that are being used. These multi-species models may still have utility outside of sister taxa, especially if we're working with groups like Cryptic or undersampled species. And combining all of this publicly available digital data, pretty much every record that I used to develop these models was publicly available through our website, through iDigBio. Being able to combine that data from the museums with current statistical methods is a really useful way for saving resources while still improving the conservation of our cryptic and undersampled species. Quick thank you to the funders and my PhD committee who helped me develop this project, as well as the 40 field volunteers that helped dig during the 5 weeks we sampled Missouri. It's a lot of effort, and I couldn't have done it without them. And I will take questions.
Thank you, Caitlin. Any questions for Caitlin?
To what extent do you think a lack of digitization in some of the collections in that region contributed to the lack of data being there? Like, it could be that there are specimens, of course, but then you just don't have the data yet.
Yeah, absolutely. So as far as I'm aware, at least the major crayfish collections in the United States have been digitized. We're doing a lot better than some other species on that front, but absolutely, there's a chance that like state agencies, Iowa DNR, might have collections that they just haven't made publicly available, and that is the kind of data we need to begin directing those surveys. So this is kind of an alternative route to figuring out where to go if you don't have access to that sort of data.
I'm curious, by the way, good talk also. It was really cool.
Thank you.
But I'm curious if, I know models are notoriously difficult to publish. What happened to the data that you collated and QA/QC'd and all that? Is it available through the American Crayfish Atlas or somewhere else where it's linked to a DOI or another identifier?
Sure. So the American Crayfish Atlas is a public website. The maps are available, but we don't have like the actual coordinates available for download because we've got some data sharing agreements in place. Most of the data that we got came from either INHS or iDigBio, which would be publicly available to download. And this data set, because I used some museum records or agency records that I'm not allowed to publish publicly, is probably not going to have a public data set available. But I'd say maybe 75–80% of the data is already out there on different websites.
And the part that is, I guess what I'm getting at is do you foresee a way that you can, coordinate all that information where if somebody wanted to reproduce your work, they could say, okay, I understand I can't get these records, but here's a quick way for me to gather everything that you used?
They would probably just have to email me.
Gotcha. Okay, cool, thanks.
Yeah.
Any other questions for Caitlin?
All right. Ooh.
So Caitlin, what can you say about what you learned about the data? Are there data points that you could think about, oh, I wish we had sampled that information or collected? Is there missing data that you could imagine wanting?
I think, apart from the obvious missing data where areas haven't been sampled, the biggest data issue we have is with collections not keeping up with taxonomic revisions. So I had to do a lot of data QA/QC for building the website, and we had to go through a lot of collections and update the taxonomic revisions ourselves. That just takes taxonomic expertise and time. Unfortunately, I don't know if there's a recommendation for broader collections other than hire taxonomists. All right. Thank you.
Thank you so much.
Okay, our next presenter. Our next presenter is Czako. She's going to be talking to us about further development of South Carolina, I presume, herbarium data entry standards to streamline data entry and species tracking. All yours.
So as he said, my name is Czilla, and we're going to be talking about herbarium data entry standards. So just a real quick intro. This is my team. I work for the South Carolina Department of Natural Resources, Natural Heritage Trust Botany and Plant Conservation Program. It's led by Keith Bradley. We've got an upstate botanist, Sam Tessel, a low country botanist, Eric Ungberg, and myself as the data specialist. And we have a wonderful team of botany data techs who process our data across the state and collect specimens. So our program goals and how it relates to this is first, we assess all of South Carolina native and present flora and give them conservation rankings. And then we track all occurrences of S1, S2, and S3 species in the state. We do this, we have a variety of references, but the main one that applies to this talk are herbarium specimens. We then use this to update our legacy Natural Heritage Trust database, and then we also conserve. So we use this data that we collect from these public-facing portals to purchase more heritage preserve land to protect these species. So on this project, we worked very closely with University of South Carolina Herbarium, curated by Dr. Herrick Brown, and the assistant curator, Avery Browning, who worked a lot on this project with me. It has over 130,000 specimens and over 20 research affiliates, volunteers, and students. And for those of you who are not familiar in this talk, a herbarium is a collection of preserved plant specimens maintained for scientific purposes. So a herbarium specimen is a dried plant, and it's glued to a piece of museum quality paper, and then this is used to preserve the specimen that can be used as a reference for research. And then the label contains the associated data for the specimen, and it tells us the who, what, when, and where. So what my group is looking for are occurrences. And so it's really important, when I say occurrence, I mean the plant growing at a specific place in a specific time, and it's recorded. Herbarium specimen are, I guess, different from biological specimens, other groups, is that you can produce a lot of duplicates. So our goal is to count the occurrences, not the records. So this visual just shows that here's a herbarium label and we're pulling it out. Issue is there are formatting on these labels, but each label is formatted by the collector themselves, so they might not be following something standard. It might be mixed between handwriting or types. So how do we get this in? A lot of our work has to do with understanding how institutions process and parse the data. So I do want to acknowledge there are data standards out there, but a lot of times you've got students or volunteers or curators making executive decisions when they don't know what's going on and where to exactly fit it into the symbiota fields. So we wanted to understand, okay, let's talk to these institutions and understand their thought process. And then we wanted to standardize the way the data is entered for at least the state of South Carolina. Although we do want to hear from other states, and we would love for it to be, uniform across all states. And the main goal, since we batch pull a lot of this herbarium specimen data from the CERNAC portal, is to train data processors more efficiently. And then especially since a lot of herbaria need more volunteers, and it's a training load for a curator or somebody else to train that volunteer. If you have a set standard, no questions, that's unambiguous, then it's a lot easier to pull in volunteers. So Avery and I prioritized this by, first of all, looking at the fields that were most ambiguous. Symbiota does have help fields, but some of them can be a little bit ambiguous. The ones that are public facing that I would actually pull, and not institution specific. So we're not worried about catalog numbers or how they put those in. And then we looked at the ones that appear frequently and are the most important for identifying unique occurrences, because that's what's really important for our program. So first, we examined how we process the data, like what do we need, and how is USCH processing the data to produce a set of suggestions. And then we pulled in as many South Carolina and neighboring herbaria as we could, and we talked to them to see what they were doing. Then we compiled all of these results into, after the discussion, into data standards. So I'm not gonna go over everything because there's a lot, but the main things I want to talk about are collector name and number, collection date, latest determination, and the country, state, and county. So the collection name is just the primary collector of the specimen. You would think, okay, great, they just put their name, but they are not consistent. So what we landed on was entering the verbatim name. This reduces data entry errors. Volunteers are not trying to figure out what, does Cindy equal Cynthia? They just write Cindy. So it makes it a lot faster. And then the names are entered in the last name, first name, middle initial, suffix format. So that just is the library format. It just assists with data cleaning. And if there's no collector name, they enter unknown. Long term, supposedly, fingers crossed, there will be some sort of tree similar to the Taxon tree, which will support synonymy. So the verbatim name not only provides context of the label, it also will help build that synonymy relationship instead of actively correcting everything. The collector number is the unique identifier of the specimen collection assigned by the primary collector. This has been used by botanists for centuries, and it's very important because together with the collector name, if they've done a good job of assigning unique collector numbers, you can pretty easily identify unique occurrences. Again, they put it in all sorts of different formats. So we suggested that they put it verbatim except for commas. That's just a quirk. When you search online on the CERNAC portal, if you put a comma like 10,100, it's going to search for the collector numbers 10 and 100. So we just ask people to just remove the comma, but everything else should be in there. Our group suggests to use s.n. for no collector number. That's because it's Latin first ad numero, and it indicates that somebody has actively looked at the record, they've tried to find the collector number, and the collector did not provide one. So it's not just left blank because the volunteer just didn't get to it or didn't know what was going on. However, there's an issue if you do the duplicate cluster search on the administration panel in CERNAC, it will link s.n. It counts it as a collector number, and so it will link it as duplicates. But that's okay. You can contact Symbiota Help. They can delete the duplicate clusters that have s.n. on the day that you ran the tool, if you choose to follow these standards. Collection date is the date the specimen was collected, and again, it's written in all sorts of different ways. So this we chose, there are programs, and there are many variations of programs that can decipher all different date formats. However, we chose to go ahead and standardize it just to reduce confusion for the data processors. So that way they don't ask questions like, is this date okay? Is it fine if I add it in here? We just tell them point blank, just add it as year, dash 2-digit month, dash 2-digit day. And then sometimes people do put ranges. And in that case, we suggest that you add the earliest date into the date field, and then there is a place on Symbiota where you can add the range, or the full range. Latest determination, this is also really important for our group. The specimen's determination can change over time as botanists go into the herbarium and they examine the specimen. The determinations are saved in a separate table, but we do want the data processors, especially if they're trying to quickly get data in at first, is to add the latest determination. That's what we are focused on, and that's what a lot of groups, research groups are focused on, and also that is how it's going to be filed in the herbarium. So if volunteers are adding the original determination, then it's going to be kind of hard to find the cabinets. And finally, the country, state, and county. So this seems fairly straightforward, but again, people add CO dot, so we're just adding the names exactly as they appear in the Getty Thesaurus as per Symbiota. Non-matching names are added to the location remarks, and the Getty Thesaurus name goes into the field. If some of the collections that are older actually correspond to multiple counties or multiple states, in which case they add both, but they separate it by semicolons. Oh, so sorry, Google slide, apologize. It had multiple columns, but we've talked about a bunch of fields together as a group. These are the fields that we do not use for herbarium specimen, at least in the state of South Carolina, that we could find or understand. So if your organization does use these fields, do let us know about your methods and examples, because we are curious. And these data standards that I was talking about have already been in use. It's being used to reduce the training load of volunteers at USCH and Clemson. It's reducing the training load of our staff in SCDNR, and we're using it to serve as a guide for notes from nature. And USCH actually has a campaign that is using these data standards. These data standards we try to make them as accessible as possible. There's an audio-visual presentation, there's an index PDF, there's a printable PDF, and it is online. We try to make these standards as, to stand the test of time, but obviously there might be new information or there might be a change. So we consider the standards as up to date as when they are published online, and at least that helps us batch edit things quickly based on the date if we change something. So these are the meetings that we have held this year and everything that we talked about. And we do have upcoming meetings, and we love to hear from everybody because, again, we're trying to understand how this data is being entered so that we can focus on pulling data from it. So please let us know if you want to be added to our email list. And our feature work includes beginning of the semester workshops so that universities who rely on student volunteers can actually attend. So again, that reduces the training load for the universities and increases digitization because then curators are more welcome to the idea of students. So thank you to all of these people. We had so many people from South Carolina and neighboring Herbaria help us. And of course, Katie Pearson and Symbiota. So yeah, you can access our resources.
Thank you, Csilla. Any questions?
Hi, I was just wondering if we use authority files for the collectors. There's the Harvard list. Have you got an option for them to select, because they're all spelled differently and all, they're all slightly different formats. Do you use the authority list at all?
So there's not a drop-down list for the collector, but we do reference it frequently. So in the SCDNR and our data, when we pull the data from CERNAC, we do use the Harvard database to kind of sort of standardize the names as best we can.
Hi there. Thank you so much. I, as an undergrad at the Arizona State University Hasbro collections, I actually spent a lot of my time digitizing the collections. Mm-hmm. One thing that I came across after quite a bit of training was that I had introduced a fair significant amount of errors into the data, because even as a somewhat trained undergraduate student, I didn't quite know how to do everything. Mm-hmm. And even now, I know, looking back on the few thousand records that I've updated, there are records that I just know have errors in them and I have still yet to correct. How much work has been done into looking at the introduction of human error into this and the effect that that has on the trust of collectors in having their collections digitized?
That's an excellent question, and I come upon this all the time. Because I started as a data processor, and so I too, I'm looking back, I'm like, I didn't know. So, I mean, I view this data, it's not a one and done. I mean, every single time we do this, we think, yes, we're just going to have this complete data set, we're going to go through it completely. But it's still better to have something on there and then to go back and clean the data. And actually, the more you go through it and the more you clean it, the easier it is, because there is somewhat of a slowdown. So USDH is 130,000 specimens, right? Eventually, we're going to get through, and we're not getting 130,000 specimens every single year. So there is space and room in the future to go back and double check to see if the records are actually matching the standards. And there are programs that you can run and create to see or to look for inconsistencies. But that is an excellent question. Oh, sorry.
I have a very specific question. I was very curious actually about how to deal with date ranges, because I've always been really curious about how other people do it. And because whenever I'm converting them to the ISO format, I just go with the last date and you go with the first date. And I'm wondering, is there a common practice? Is there any standard about, do you use the first or the last or something in between?
So it's the first. I mean, that was what we went with. It was the earliest possible date, and that's because we're always, from a species tracking point of view, we're thinking, when is the first time it was seen? So I know exactly what you're talking about, because some people will add a date for when it was flowering and then when it was fruiting, so there's a date range. But we are interested in the earliest date that it was seen, so that earliest occurrence. But I do also try to grab the secondary occurrence too as a follow-up, the plant is still there. And there actually is, and I'm sorry I went through this quickly, but I promise you everything is online. There is this little button where you can add the full range. Mm-hmm. 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 Yes, you can. So that is a wonderful feature of Symbiota is if you have a user. Some herbaria create a volunteer user on Cernac, which I highly recommend you do not do. Give everybody their own account, and that's not to single anybody out. It's just to like help follow up. I've also found that having consistent meetings with the data processors really helps as well. But I'm out of time, so. Perfect.
Okay, we're going to have to move on, folks. Thanks. All righty, staying on the herbarium theme. And the South Carolina theme, yes. Our next speaker is Hannah Berberich, who's going to be talking about a novel Cryptogam herbarium specimen imaging procedure prioritizing both specimen and label.
All right, hi everybody, good morning. Like you said, my name is Hannah Berberich, and I am here with the South Carolina Department of Natural Resources Natural Heritage Trust Botany and Plant Conservation Program. It is a long name, but we do a lot of important work, and we're really proud of it, so I'm going to talk about some of it. This is the novel Cryptogam herbarium specimen imaging procedure that prioritizes both specimen and label. So this is our team. We are led by Keith Bradley, who is our state botanist, Samantha Tessel, who is our upstate botanist, Eric Unberg, who is our low country botanist, and Czilla Czako, who you all just heard from. She is our botany data specialist. And then on top of that, we have a bunch of botany data techs across the state who help us process data and collect specimens. So we have 3 goals at our program. These are going to be to assess, track, and conserve. Through assessing, we are looking at all of South Carolina's native flora, and then we add conservation rankings based on nature serve criteria. Through that, we track all S1, S2, and S3 species, and then we use information from collections and from herbarium specimens in order to conserve and acquire properties that we're interested in that have rare species. So you might be wondering, what is a cryptogam? Not everybody knows this because they are, tiny and minuscule. We don't always notice them. But they are going to be the 3 subkingdoms of Thallophyta, Bryophyta, and Pteridophyta. Today we're going to be focusing on, oh no, I didn't do it. Well, today we're going to be focusing on the lichens in Thallophyta and Bryophyta, which is mosses, liverworts, and hornworts. So Bryophyta, which is the mosses, liverworts, and hornworts, they're small, non-vascular, non-flowering plants, and they reproduce via spores, and they lack true roots. Thallophyta is lichens, and they are the symbiosis of algae or cyanobacteria that lives within the cortex of fungi and creates a mutualistic relationship. Cryptogam labels can be, here nor there. They vary depending on the collector, and they can include a lot of data or no data at all. So through imaging the labels, we're going to be imaging the original label, and then at the University of South Carolina Herbarium, we created a new curatorial process where they get new typed labels. So through that, you have the original label that has all of the markings, indications, anything the collector would have used to track their collections, and then the official label that's easy to read and typed. Imaging the specimens was a major part of this process. It is important for analyzing community structure, the health of the specimen, as well as any taxonomic changes. So if you have a specimen that you're not sure what it is, you just put a genus on it, other people can look at these images and possibly identify it. The amount of specimen material is also a very important part of this process. So through imaging the amount of specimen material, you can look at it online and not have to open the packet with the specimen in it. So this way you're not degrading the specimen through, opening the packet, reclosing it, taking it out, looking at it. You can just look at it all in the same place with the label. You can also analyze reproductive structures if they are available and if they're, still in good quality. But this is even easier when you do it when they are still fresh. So you can look at the microscope pictures that are associated with the collection records and even look at them in the dried specimen photos as well, but preferably microscope pictures. Analyzing the community structure is also very helpful. You can determine the health of the environment that these plants are in, as well as any associated taxa that are nearby, and the substrate that they are on, so you can determine any symbiotic relationships that exist and community health. So the imaging setup, which was my favorite part of this process, my dad helped me create this 3D printed plate. And basically it has 3 sections like a lunch tray, so you have all of your different components separated. In the top left-hand corner, you've got your specimen in a weighboat. Each specimen gets a new weighboat so that no genetic material is compromised. And then in the top right-hand corner, you have got the new label on the new packet. And in the bottom right-hand corner, you've got the old label that goes on top of the specimen when it's put inside the packet and folded up. Through this, you can zoom in on the specimen images. It's not super high quality, but you are able to tell some diagnostic features. You can look at leaf shape, the shape of the specimen itself, the amount of specimen material like I mentioned, and then reproductive structures as well if they are available. And then you can also zoom in on the labels. This is especially helpful for the old labels because you can zoom in and see all the little markings. Like I said, some collectors use shapes, they draw little symbols, that kind of thing. So those can be diagnostic in determining where these specimens were collected as well. The imaging template, it's lasered onto bluestone silicone, which is a liquid stereolithography resin, also known as SLA. I painted it matte black so that the glare from the light box doesn't, harm the integrity of the photo. And then, like I said, this allows for easy imaging. So when you have all the sections, you can just put everything in, image it, and go. It's a lot easier for volunteers because you're not having to reposition everything every single time. So it's just more time efficient in the long run. Microscopy is also a large part of this project. It was important for us to have these images associated with image tags on Bioportal. So that way you're able to look at the image of the specimens on the bryophyte plate as well as on microscopes, and then being able to tag those images with their specific indicators. These were the image tags that got added to Bryophyte Portal. I'm going to go through them in a little bit more depth. So stacked focus images are images that are taken at varying degrees of focus. So that way you have a more comprehensive 3D view of any anatomical structures. And this can be applied to more than just, you can have more than one tag on each photo. But yeah, these are really cool. They're my favorite type of photo. Compound microscope images, these are going to be taken on compound microscopes, almost always on a glass plate. That way you can amplify any small anatomical structures like oil bodies within liverworts. Dissecting microscope image is going to be more of a 3D view of plants, not like the folk stacked image, but yeah, they're going to be on a compound or dissecting microscope, not on a glass plate, but that way you can amplify some of the small features as well. This is really helpful for looking at leaf edges as well. Field image, this is ideally going to include some of the surrounding area where your plant is, and the substrate as well, and any associated taxa. Macro photo, this is also going to amplify some of the small parts of your plant, and these can be taken in the field, fresh, dried, or under a microscope. Sexually reproductive is going to include sporophytes and gametophytes on acrocarps and pleurocarps, and this is not to be confused with vegetative propagules, which are part of bryophytes that can be broken off and grow a whole new plant. These include leaves, rhizoids, and stems. And then utilizing image tagging, like I said, you can apply one or more than one image tag to each image associated with the specimen. This allows for easy sorting of the specimen. So if you need a stacked focus image, you just want to look at one. You can go through, look at all the stacked focus images on Bioportal and look at how cool they are. This is beneficial for studying as well. So if you're trying to learn the reproductive structures of a specimen, you can put in the sexually reproductive tag and you can look at all the photos that are on there. How to search them, it's fairly easy. You just want to click on the images tag on Bioportal, and then it will come up with a drop down. You can select any of the image tags that are available. They're more available than what I mentioned today. What I mentioned today is what we got added. And then once you select which one you would like, it brings up all the photos associated with it. And it's really cool that people are already using it. I was really happy to see that. This is the field image tag, but you can apply more than one as well. Then thank you to all these people. They gave me very helpful feedback and helped me with this project. And then if you have questions, comments, or suggestions, let us know. Feel free to scan the QR code for my contact information.
Thank you, Hannah. Any questions?
I was just wondering if you have documentation and like workflow protocols for what you proposed with this.
For which parts?
I would say all of it. I guess I'm curious especially about the tagging of the images and then what parts you have the volunteers doing and then what parts you have yourself doing in terms of integrating all of the different image types into one record.
Yeah, so we don't have a ton of images up. We've only imaged 8 specimens with the bryophyte plate. The ones that I did do for that, they did get image tags applied. But anyone can do it, and it's easy to go in and add as you're adding in the specimens. There's a little drop-down box for it. Not on me, no.
So the cryptogams can be difficult, because they often have like different depths of fields. How do you manage that in your images and with like the lunch tray kind of setup that you have? Like, has that been an issue that you've come across so far?
No, not really. So that's why we added in the image tags. So the bryophyte imaging plate is more of a standard for all of the specimens, mostly to be able to see the amount of material we have to work with if you want to do destructive sampling. Then you could go through later and do, a stacked focus image and add that in as well, but the template is more of a standard for everything.
Any more questions? All good. Okay, I think we'll move on. Thank you.
Thank you.
Alrighty, our next speaker is Tommy McElrath, who's going to be talking to us about using TaxonWorks visualization tools to improve data quality at the INHS insect collection.
Thanks, Andy. So I actually have a little bit of a title change. This was what I originally submitted, but after some conversations at TaxonWorks together, I'm actually going to change this from quality to fidelity, just because there's a lot of subjective ideas about what data quality actually means. And when we're talking about data fidelity, more talking about how true we are to representing data digitally versus what's on the specimen. So I'll be referring to that instead of quality throughout the talk. Just to give you a quick background on where we're from, this is some specimens from the INHS insect collection. We're in Champaign, Illinois. We have about 2 to 3 million pin specimens, about 150,000 slide-mounted specimens, about 4 to 5 million specimens, both in lots of alcohol as well as individual vials. We've been digitizing for a long time, with many different groups being 100% digitized, but a few groups with massive backlogs of specimens left to do. Represented, we have about 1.1 million collection objects digitized right now, representing about 2.7 million specimens out of an estimated about 7 million, but that number's growing. And we've been digitizing since the 1990s, and for an insect collection, that's quite a long time. And with that, we have a lot of problems that come with the question of data fidelity. How well was data represented digitally back in the 1990s versus now? And we also have a lot more research questions that we're asking now that were not being asked in the 1990s or before. And if we're going to represent that data digitally, standards have evolved quite a bit since then. We do export our data to GBIF and IDIG Bio and use all their wonderful data quality flags on there. And once we started exporting our data to GBIF more recently, after a long gap as we migrated from a series of legacy databases, we noticed a lot of issues popping up with their data quality flags. Things like coordinates being rounded, continents being derived from countries mismatching, taxon names not being found. We started to run into the problem of how do we track both issues from the legacy migration as well as the actual issues with the data fidelity itself. How do we keep track of this? So originally, as we were doing the migration from what was a FileMaker Pro, Microsoft Access, and custom SQL databases, we used a GitLab tracker to start to try to track different data problems we were having. This was private and internal, so just pretty much for us, which led to some issues with sharing it and actually getting people to contribute to it. And so you can see some of our issues here, everything from things not having geographic areas or certain objects just not being able to be loaded for some reason, all the way to fixing cross-project issues and things like that. So we decided recently to actually move this to GitHub to make this more of a community resource, as we've had some people contribute to helping improve our data fidelity recently, and we couldn't do that with a private tracker just because it wasn't out there and it wasn't easy to actually start to get that data visible so people could start to contribute to it. For example, this is a recent issue that was opened by Bob Mezabov, who found 71,000 of our records have no entry at all in scientific name in our export, and so those obviously are not being searchable, and so we need to start to figure that out. And as you can see here, I've started breaking this down using some internal tools. So what are we doing to actually manage our data? We've migrated all of these databases into a TaxonWorks database. And we have a lot of tools right now to get data in. We have a cool grid digitizer to get slides in. We have some really fun ways to break down individual data on grids and then parse it individually. We have a Darwin Core importer to import a lot of different occurrences that already have spreadsheets associated with them quickly. And we also have a lot of verbatim fields in order to increase data fidelity by not just parsing fields, but also preserving verbatim data that's on specimens. And all this gets exported to the new Darwin Core term verbatim label. And we also have verbatim fields for individual collecting events, and those get exported to the appropriate Darwin Core terms as well. And then we also have data attributes that are on every single specimen that was migrated, and so this is just a duplicate level of preserving data fidelity when we migrated from our old databases, just to make sure we did all that. So let's go over a few of the issues we found that we started to track and start to fix with some newer TaxonWorks tools. For one, we found that historically we were tracking biological associations as one of the coolest part of our data. But unfortunately, almost all of them were completely reversed with the subjects and predicates backwards. And so we went through and had to fix those. That was initially going to be through some code, but new tools in the filters have actually let us do this manually, where you can select any number of associations and change or flip these associations as you want. So I was able to do this as a collection manager with no code required in only a couple days, just churning through these very quickly. In TaxonWorks, we also have this idea of geographic areas. Instead of individually parsing out country, state, and county, you just select a geographic area from a pre-populated gazetteer, and that's automatically put in there for you. And you can search by those spatially or via coordinates or that sort of thing, unless your geographic areas that are assigned don't have shapes. And these were only imported, I think, as a accidental, or not an accidental, as a backup for things that just don't have any shape files that are publicly available. And so initially, all 8,000 of these uses for the state of Illinois, for example, were imported to one that had no shape. And so when you did try to search spatially for things that occur in Illinois, these weren't coming up. And they also weren't exporting properly for some reason. So there is now a radial mover, assigning a mover to go ahead and do that for specimens. So I went ahead and did this for any objects that didn't have shapes or any collecting events that didn't have shapes. I moved over 10,000 records, also pretty quickly. One of the coolest new tasks that we have is this project vocabulary task that Debbie Paul, who's our community liaison, helped the developers come up with. So you can search pretty much any of your data classes or things in your project. For example, this is a filter for method, and since we're an insect collection, apparently we like malaise traps. Along with a lot of other methods for collecting. And so you can actually go through and click these different filters, and it'll actually click on any of these different words, and it'll actually send you to a filter that shows you all the objects that have that word cloud in that data class. So this leads us to do a lot of cool data visualization things. For example, if you want to synchronize all your uses of ultraviolet or blacklight traps, you can go through and do that using another new task called Field Synchronize, where you search for any sort of attribute or predicate, and then you can bulk edit and change all those things across your project. One other really useful task is a new task called Stepwise Determinations, where we take verbatim strings of determinations and you can parse them in bulk to OTUs, people, and years. And so this way I was able to pull through thousands of different determinations on specimens in bulk instead of doing them one by one. Along with the help of some of my hourlies. Really the core of all these different data visualizations is our filters, which basically let you find a set and do things to it. For example, this is a pretty complicated query of collecting events that don't have georeferences, don't have a collector role, and with attached collection objects, but that do have a value in verbatim collectors. So these all need a collector role, but don't have one yet. And so I can go through and start curating this data set. Other kinds of things you can curate in these filters include setting your verbatim fields in bulk, moving or assigning those geographic areas, setting your collection date and time, or adding new collector roles. And these are being expanded with further improvements soon. You can also send things to a different task called Collection Object Match, which is slightly differently in that it basically lists a series of catalog numbers or identifiers, and then you can do all sorts of other additions, things like preparations, repositories, tags, added bulk determinations, collecting events. This is also just different ways of doing the same thing in some extent. But notably here, you can just list a set of identifiers and do a whole bunch of things with them. So for example, I modified over 10,000 specimens that had no prep type and added the prep type using this task. And in combination with collection object filter. So let's put it all together. Using our GitHub tracker, I started a sort of mini project to get rid of our unknown collecting event that was a complete artifact from our data migration. So I created an issue in our GitHub tracker where there were over 20,000 collection objects on this unknown collecting event, which was basically an artifact from FileMaker where if someone didn't know what something was, they put it as unknown. And so we migrated. We obviously couldn't create brand new collecting events for all those, so we also just put it on here. And obviously, 20,000 collection objects isn't that many, but it looked like there were tons of actual good data in here. For example, we had over 3,000 objects from Charles Robertson's bee collection, which are represented by numbers, and so hourlies didn't know what to do with those, either because of poor training or something else in the behind the scenes, but we managed to get those transcribed as well. So initially we started doing these one by one, but then we started breaking them up by queries, and I will note this represented over 20,000 specimens. This is even after lots of fixes. We still have 11,000 to go. So we've already churned through about probably at least 20,000 specimens with new data added just by starting to parse these things. By tracking these filter queries in GitHub, we were able to break it up, and just thanks to Lily Hart and Michelle Kohler for helping me do these. So you can see we've already finished the ones that I think were Charles Robertson records, some other ones from different sub-projects as well. But we still have 11,000 that are probably linked to our historical accession records, which were also represented by a single number. So how do we deal with those? I downloaded these as a Darwin Core archive from that same filter, opened these with OpenRefine, sorted them by one of those data attributes that was put together for data fidelity when we migrated, grouped the groups, and actually started bulk churning through these by sets instead of individually. So I was doing 44 at a time instead of 1 at a time. I sent these to collection object match, and I was able to churn through 5,000 collecting events in less time than it took 3 people to get through 3,000. And so you can really get through and actually start to improve data fidelity using these tasks. So where are we now? I'd say we're kind of lurching towards data fidelity. There's still tools that are being improved or developed. And I really want to emphasize that if you give collection or data managers the tools to actually see where the problems are, they will start to be able to address them. But you have to give them those tools and not expect them to create things like code or custom processes that they don't know how to make. If you give them the tools, they can do it. And I will say also that as the community starts to curate things, it leads to a lot more actionable tasks that I can do. So community creation is better. Putting your messy data out there is better than hiding it behind a paywall or a user login. So if you want to hear more about TaxonWorks, we're going to be at the data cleaning workshop on Friday, myself and Debbie Paul, or you can always go to TaxonWorks.org and get started from there. So thanks to my hourlies, the species file team who writes TaxonWorks, and any people who've been contributing to our data fidelity. Thank you.
Thanks, Tommy. Any questions for Tommy?
None? Tommy, what database were you familiar with using before TaxonWorks?
I was using Symbiota, mostly.
Yeah. So I'm curious with people in here, and Tommy, your experience with your ability to visualize your data. Like, what do you have to do to discover the issues with your data? And so, Tommy, what's your, I mean, I heard what you said in there, for example, but I'm wondering if you're already beginning to think of other ways in which you could see things in your data or see patterns that could help you. Use the data for research as well, obviously.
Yeah. So I didn't have, initially when I was using Symbiota before, I was the one putting all of it in to begin with. And in this case, I have been trying to visualize all the historical artifacts from our data from the past 30 years. And so it's very difficult for me to have any ideas without any of these tools. And so I don't have too many more ideas, but I'm loving what's going on right now.
So to that point, I want to add to Tommy's comment. We all have this sort of knowledge. It's our local knowledge. And we know it, but we might move on to another place. We might move to a different database. That knowledge goes away. How long does it take you to get to know your data? When you move to a new system, or when Tommy hires a new student, or how do they get to know what's actually inside that database?
I don't know, because it's taken me 7 years to get to this point with our data. I've started to put together resources for people to learn things so that students can, for example, know that there are already pre-populated collecting events for every single one of our accession numbers that they can just grab, instead of just saying, oh, this is unknown, I have no idea what to do with these, or typing in the number, or trying to redo everything. So at the very least, you can start to actually document all these processes that would lead to these bad data fidelity things in the past.
So to that point, how many of you have done minimal data capture or skeletal data capture in your digitization efforts? How many of you have done that? Right. How many of you have a plan for how to go back and fill in the missing data? And are you actually doing it? Okay, cool. So this is another way in which my question comes here. If you move on, who else in your project knows your data and what got filled in or not? And how are they going to know what's still empty or could be filled in? How can they discover that?
One thing I would love to see is the MIDS data standards basically applied to all my specimens, and having some sort of visualization tool to see, hey, these things are MIDS level whatever, or these things are not. That would be a great tool to go on from there. Minimum information digital, minimal information for a digital specimen? Is that what it is? Minimum information data standard, that's it. Look at the TADWIG website. I think we're out of time, so thank you.
Thanks, Tommy.
All righty. Our next speaker is Brooke Long-Fox, who I don't think is here. was going to present online. I do not see her online, but I have a recording of her presentation somewhere. There it is. Hopefully. Or not.
Thank you for attending, and I'm happy to be presenting virtually this year at the Digital Data Conference. I'm sorry I couldn't be there in person, so I thought
there we go. Technical difficulties. Hold on 1 second. I'm getting feedback. Yeah, it's coming through there. Let's do this.
Hi, everybody. Virtually. I'm sorry.
Sorry. This is all.
Virtually. The MorphoBank director, but we're happy to assist in any kind of data formatting or publishing. MorphoBank projects are associated with 6 different sources. These are various journals, and some of these I've listed here are top journals: the Journal of Vertebrate Paleontology, Cladistics, Zoological Journal of the Linnean Society, Journal of Systematic Paleontology, and some of the American Museum of Natural History journals. And so MorphoBank has data associated with articles in these journals. And often, most of the journals on MorphoBank are behind paywalls. Very few of the publications are open access. So when scientists add their data to MorphoBank, they are actually allowing people access to the data that otherwise you might have to pay for to access the article and see the data. So we are providing an open access and fair resource. Through MorphoBank's website, you can also build and access morphological matrices, phylogenetic matrices, and use tree building software. MorphoBank has over 1,600 projects. This increases every year from researchers adding their own data or curators adding data. Of these projects, 382 are NSF funded. These projects are from all over the world, so there's other funding sources that also support this project, but MorphoBank has been heavily NSF funded over our close to 25-year history. In these projects, they represent over 200 peer-reviewed journals. They also represent 20,000 species. There are over 1,000 phylogenetic matrices. These can be downloaded and reused for additional scientific research. There are over 170,000 scientific images. These are mostly of different taxa or various pictures of appendages or parts of different organisms. Over 3,900 scientists use MorphoBank, and there are approximately that many visitors each month. MorphoBank allows matrix building and tree analysis. So for example, you can either start from scratch and build a matrix within MorphoBank, or you can upload a matrix from a TNT or Nexus file that was created in another program. You can also search for and download matrices that were published in MorphoBank, and then use the previously published matrix to build upon for your own research. Once a morphological matrix is completed in MorphoBank, this basically means that all the taxa have been scored in the character matrix. You can use the matrix for phylogenetic analyses. Some phylogenetic analyses use significant computational resources. So if a user does not have those resources available, you can do it within MorphoBank. MorphoBank has the capabilities to analyze the data without leaving MorphoBank. Currently, we do this through a partnership with Cypress Science Gateway, which is a public resource for inference of large phylogenetic trees. MorphoBank has access to phylogenetic analysis under parsimony using PALP. And so that is a parsimony-based method. So in MorphoBank, you can select the matrix you would like to use, and then select any kind of special model parameters associated with the parsimony analysis. And then without leaving MorphoBank, the matrix and the parameters are sent to Cypress. and a phylogenetic tree with any related statistical data is returned back to MorphoBank. We are currently working on an expansion with Cypress to include Bayesian analysis. This will probably be Mr. Bayes or Beast. And we're also working with the people at TNT to get a connection similar to what Cypress has, but with TNT to allow additional parsimony methods. So how is MorphoBank data used? MorphoBank has been around for over 20 years, so we've recently been trying to quantify where the data is going, how are scientists using the data, and so we've recently done a Google Scholar search and tried to quantify what MorphoBank data is being used for, and what we're mostly seeing is that it's being used for phylogenetic analyses, describing new species, and examining the diversification of taxonomic groups. In terms of taxonomic groups, it's a wide range of organisms, mostly vertebrates. This is including things like dinosaurs, reptiles, mammals, and there's even a couple studies on human evolution. Less commonly are plants, invertebrates, and microorganisms. So we have a couple data sets that are related to different things like diatoms and small organisms that can only be seen with a microscope. So MorphoBank is always expanding, and we're looking to improve MorphoBank. And so any feedback or suggestions for program parameters would be great. We are also looking to integrate non-morphological data sets. So that will be looking to apply for some more NSF funding to integrate more data that's not supported in other databases. So if you have any suggestions, or you would like to work with us to integrate any other type of data, feel free to email us at curator@morphobank.org. MorphoBank has received funding from NSF. This funding most recently is by NSF, DBI, and EAR. So these are to build additional computational resources for phylogenetic trees. This is like adding parsimony and Bayesian algorithms. Also, some of this funding is to support the replatforming of MorphoBank's current software. It's currently running on over 20-year-old software, so it needed a complete overhaul. And we are also updating and redoing the website to make it speedier and more user-friendly. And in addition to NSF, MorphoBank is also supported by different institutional memberships. These are all listed here, and we would like to thank all of these sponsors. If you have any questions, please let me know, and thank you for letting me attend this conference and letting me present virtually. Thank you.
No, it's
within Specify. So my background, I am not a biologist. I just graduated like 3 weeks ago from my master's program in information science. So I was coming from this with a background in libraries. And the scope of the work in the summer of 2023 was to take M-PABI from nothing. We didn't yet have specimens that would be accessioned into the collection, although those were collected during the summer. And we didn't have a database instance set up. So the goal was to take it from nothing to launching that first iteration of the database. And for this talk, I'll walk us through the different steps that I was directly involved in that summer. So our first step was to determine what fields we would need to accommodate in M-PABI. Because of the nature of the pathogen biorepository, we would be accessioning specimens from such a wide taxonomic range, we needed to understand what we would be looking at. So we began by contacting researchers in various research areas like mammal-parasite interactions, viral surveillance, live fungal cultures, and the other areas listed here, and asked them to give us examples of their field data. And those came in various forms. Sometimes they were just the empty data forms that they filled out. Sometimes it was spreadsheets, other things. And we took that data and mapped it to Darwin Core initially, just to begin to get a sense of what sort of data we would anticipate accessioning in M-PABI and where it would need to go. At this step, we also took into account some of those fields that wouldn't necessarily be in a researcher's field data, but we would want to see in the database, like those verbatim fields in Darwin Core. Next, we analyzed those mappings for redundancies, or even sometimes talked to researchers to better understand whether data was collected for a really specific purpose for their research project, and that wouldn't necessarily need to be reflected in the database. So we identified things like, here these two different sources are two different ornithology projects that were conducted by two different researchers, so we could look at overlaps in, okay, they're doing research in the same taxonomic area, they're doing it differently, collecting data differently, but here's a redundancy, so we can anticipate, of course, we would need latitude data. So we then standardized attribute data fields. This is a challenge that I'll talk about throughout the process, but one of the things that we saw was that researchers might be collecting the same sort of data but storing it differently. So for example, these two ornithologists, one of them collected breeding condition data in a single field, which is indicated in the blue square here, called breeding condition, that might have values which are indicated in green, like coacal protuberance or brood patch, or the field was left blank to indicate a presence or absence of those breeding condition markers. Whereas the other researcher approached this with two different fields called brood patch, or CP for coacal protuberance, and the values would be yes, no, or left blank. So following looking at, for example, the ornithology database at the University of Michigan or other comparable collections, we had to make decisions about how we would standardize that attribute data. And then we map those from Darwin Core to Specify. So this is an initial mapping before we actually began to play around in the toolbox and build out the database to just begin to see where we might map that data into the Specify schema. So one thing to note here is that we mapped that breeding condition initially into the collection object attribute table, planning to use one of the text fields and co-opt that for being the breeding condition field. So this is our initial mapping. And before we actually began building out the database, we had to think about the end goal of how we wanted users to be able to access the collection. And one of the challenges there is that we needed to find a way to store theoretically limitless amounts of attribute data. Because of the wide taxonomic range of a pathogen biorepository, we could anticipate seeing, for example, the attribute data points that would be collected for a bird specimen would be vastly different from the attribute data collected for a bat. The number would be different. There wouldn't be very many overlaps in what the actual attributes were themselves. And that would look vastly different than like a fungal culture. So the problem was that there's a limited number of fields available in the collection object attribute table. And the solution that we came up with was to build out a sort of miniature data form within collection object property. And the way that this works is this classification field here, the attribute type is a drop-down menu, so a controlled vocabulary of those different names of the fields like breeding condition or gender, things like that. And then depending on what the attribute is, the person entering data can either select from a new drop-down of controlled values for that field or enter a numeric value in the appropriate unit. And then of course we store the verbatim text and have a field for remarks. So what that looks like for any given collection object, which might be any sort of specimen or culture or anything else, we might have attributes 1 through n. So for example, maybe the attribute is reproductive condition, and we have a controlled value of lactating. Or the attribute is weight, and this isn't controlled, it's an entry for the weight, but then the unit marker is controlled. So these could be potentially an unlimited number of attributes for a given collection object. So that was kind of our solution to that challenge. And then towards the end of the summer, we developed the initial database design. And I just wanted to take a few minutes to walk through that. So of course we would start with a collecting event. And for each collecting event, there would be any number of collection objects. Something that we talked about and had to make a decision about was how we would deal with the collecting events for the pathogen. And when I say pathogen, I'm also talking about pathogen, parasite, etc. That was identified on or collected from whatever the host specimen was. So one option was to create its own separate collecting event where the date associated with that event would be the date that it was found on or identified from the host. But the concern with that would be that it would artificially date the time that the pathogen was removed from its original environment. So the approach that we took was that hosts and parasites would share the same collecting event with the same collecting date, and we would store the date that the parasite was identified in the preparation date field. So collecting event underneath it are the collection objects for the hosts and their pathogens or parasites. And then we have each of the preparations for each collection object, which of course are stored in the preparation tables and Specify. And we wanted to make those relationships between the hosts and their pathogens explicit. So we used the collection object relationship tables, and we used the OBO relationship ontology to name those relationships. So with a bat collection object, we might have the bat fly, as Kelly was talking about, and the relationship between those would be explicitly parasitized by and parasite of. You can see the different relationships here. One that we chose was simultaneous with when we want to indicate that something was collected with something else, but we're not necessarily making an explicit claim about the relationship between the objects. And then finally, we have explicit relationships between the preparations that identified the pathogen on the host. So for instance, the DNA preparation of the bat would be the parent preparation of course, the same DNA extraction that was used to identify the pathogen. And the way that we stored those relationships are to use a field in the preparation property table to store the accession number of either the host or the pathogen, depending on which one we're looking at. And that takes us to future directions. One of the challenges of taking that approach is that that's not necessarily, it doesn't necessarily translate into an intuitive way to search the database for those relationships. So future directions for the database are definitely a more intuitive search phase with things like improving searchability by providing predefined queries that we have seen people attempt to search before. So I'll stop there. Happy to answer questions. Here are a few of our funding sources. Thank you.
Thank you, Summer. Any questions?
Is it possible for me to use this software for my own host parasite data, or is it just for your collection?
That's a question for Kelly. I know that M'PABI is accepting specimens or records. I don't know if Kelly wants to address that.
Like,
yes,
and
of course.
I meant, like,
yeah.
Could I host this database, like use it myself, like the software, or could I only contribute data to your database?
Well, it's built on a specify instance, so you could set up your own, yeah.
Do you want to comment on that, Kelly?
Oh, sorry, I threw that at you, Kelly. I think I answered the question. I don't think I made it clear that this is just we just built an out-of-the-box specify database and came up with some kind of creative solutions to the particular concerns that we had with our data.
So a quick question, and this is something we're working on at TaxonWorks. So we have host parasite, host pathogen relationship biological associations. Mm-hmm. So you're a researcher who studies mites that live on bats. And it turns out that if you want to ask that taxonomist to identify that mite, you need to know not only that there was a mite and a bat, you need to know where on the bat that mite was located. Mm-hmm. They like to live in ears and warm places when it's cold outside, and the mite likes to move to the wings when it's hot outside. So you also need to know when, relative to like the hemispheres, right? Mm-hmm. So you need to know. So could you do not just the host record and the parasite record or the pathogen record, could you also do this added information of sort of when and more specifically where?
The host was collected.
Where was the parasite found on the host?
Yeah. I think there's certainly capacity in the database structure to store that information. I think the concern always comes down to how we could enable that search and the end result.
Sure. And yeah, UI are challenging for sure.
Yeah.
Hi, quick question about relating host and parasites where you have one half of the relationship, but another institution has the other half.
Yes.
I wonder how you handle that.
Yes, that's definitely something that we talked about a lot, not even another institution, but a case that will come up at M'PABI is that the host is deposited at one of the taxonomically organized data collections that already exist at the Museum of Zoology, so like the ornithology department or whatever, and the parasite is deposited at M'PABI. And the current solution for that is to just store the accession number for the host in, I can't remember what table it's stored in right now, but it's just stored in a text field in M'PABI, with the goal being to provide a URL that links to the record for the host in the other database. But that's one of the things that we were talking about as maybe potential future directions would be creating either an out-of-the-box or using a different solution to provide searching that searches across those databases. That's difficult to integrate within Specify and the Specify search UI itself, but maybe a third-party opportunity to artificially link those databases together, at least on the user end. But that definitely does not exist right now for us.
Any other questions for Summer? All good. Okay, thank you very much, Summer. So now we're going to transition into the lunch break. As I mentioned, the food truck is going to be in the parking lot across from this building. So head out there, grab some lunch, and then you can bring it back here and sit somewhere and do whatever you need to do. And then we will reconvene at 2 o'clock with Mason Heberling's discussion session. So